Hello friends. Welcome to Lambda Geeks. My name is Abdullah Arsalan. I have 8 years of research experience in biotechnology. Thank you for connecting with us. Keep watching, stay connected. Hope you are doing good. Let's start our today's session. Through this video I am going to explain the overview of purine biosynthesis, in which we are going to cover purine structure, its atomic composition, biological functions, pathways of purine biosynthesis. Let's talk about purine structure. They are heterocyclic aromatic organic compounds that consists of 2 rings, pyrimidine and imidazole, fused together. Purines are generally water-soluble, and they are the most commonly occurring nitrogen-containing heterocyclic compounds in nature. Purine is both a very weak acid with a pKa value 8.93, and an even weak base with a pKa value 2.39. If dissolved in pure water, the pH will be halfway between these 2 pKa values. Now we will discuss the atomic composition of purines. Constituting atoms of purine are assembled from a variety of precursors like glutamine, glycine, aspartate, hydrofolate, and carbon dioxide. Glutamine provides nitrogen atoms, nitrogen atom number 3 and 9. Glycine provides carbons and nitrogen atoms, carbon atom number 4, 5, and nitrogen atom number 7. Aspartate provides nitrogen atom number 1. N10-formyl tetrahydrofolate provides carbon atoms, carbon atom number 2 and 8. Carbon dioxide provides carbon atom number 6. Let's talk about the biological function of purines. Purines are predominantly found in our body. Various types of purines are found in our body, and perform variety of functions in metabolism and cell signaling as well. But in genetic material like DNA and RNA, adenine and guanine is found. Now we will discuss about the biological pathways through which purines are synthesized in our body. The pathways for the biosynthesis are classified into 2 different types, de novo pathway and salvage pathway. In the de novo pathways, nucleotide bases are synthesized from some simple compounds. The basic framework structure of the pyrimidine base is synthesized first, and then gets attached to the ribose sugar. The purine base's framework structure is synthesized in parts directly on a ribose sugar-based structure. 5-phosphoribosyl 1-pyrophosphate, PRPP, reacts with amino acids, ATP, and CO2 to form purine. In the salvage pathways, preformed bases are obtained and rearranged on a ribose sugar unit. 5-phosphoribosyl 1-pyrophosphate, PRPP, combines with purine base to form purine nucleotide. Both the salvage and de novo pathways operate to synthesize purine ribonucleotides. All the deoxyribonucleotides are produced from their corresponding ribonucleotides. The deoxyribose sugars are produced by the process of reduction of the ribose sugar present in a fully formed nucleotide. Thanks for watching this video. This was the brief introduction of biosynthesis of purines. We will discuss the de novo synthesis and salvage pathways in details in our upcoming videos. For more articles on biotechnology, advanced sciences, engineering, mathematics, physics, and various other domains, please visit our website lambdageeks.com. If you find our content useful, please share, like and comment on our video, and do subscribe to our YouTube channel.